Howdy folks, sorry I'm not in class. We still got to go over this lab. Hopefully this will help you get ready for the quiz. So here we go. Now the first thing we got to do is make sure that you don't get a 7 out of 10 because you neglected to answer one question. So go right now, check through each page, we'll wait, we can hit pause, flip through there, make sure you got them all. Especially question number 9 there on page 2. A lot of people missed that one. Some folks didn't get a chance to get all the way to the end and do the distilled water to deplasmalize the cells. That's steps number 12 through 15 on page 8. Be sure, please, to write skipped in the margin so I don't nail you for blanks when we didn't get a chance to finish it. Because some people did get a chance to finish that. And no, you are going to have to think about some of the questions. I feel like we've already gone over part 1, so I'm going to skip the part 1 stuff and go right to the part 2 questions and the analysis questions. So go all the way to the second to last page, page 8. We'll look at question number 11. This is just asking about the observations that you made after putting the red onion in salt water. I'll ask a couple guiding questions here for each one of these questions, like, well, what's the concentration gradient here? Every time you are doing a diffusion question, whether it's osmosis, where we're looking at water, or diffusion, where we're looking at some other particle that can go across the cell membrane, you have to determine the concentration gradient. So where is there a high concentration? In this case, we're talking about water. So where is there a high concentration of water, and where is there a low concentration of water when we put a cell in salt solution? So is that salty water considered hypo or hypertonic to the cell? That's the kind of the big question. And what happens to cells when we put them in hypotonic solutions? Well, cells swell when we put them in hypotonic solutions, but since the salt water is not hypotonic, it's hypertonic, then it's going to do the opposite. Then the cell is going to shrink. Alright, so hopefully you saw the red cytoplasm kind of pull towards the middle as the cell shrank and the cell wall held its shape. Alright, moving down a little bit on the same page. Remember to write skipped if you skipped the 12 through 15 here. So here, surprisingly enough, we have a student or a group of students who have not followed the directions. Can you imagine? Oh. And so they reversed things. Instead of putting the starch indicator solution on the outside, they put that on the inside. And instead of putting the starch and glucose on the inside, they put that on the outside. So they got everything reversed. So of course, that means basically their results are going to be reversed. But let's go through this a little more carefully. So in our cells, where did the color change? Inside or outside of the cell? And more importantly, why did it change inside of the cell and not outside of the cell in our situation? When we follow directions, why did it change inside and not outside? Right, that's because the iodine is able to go through the cell wall, or excuse me, the plastic bag, but the the starch is not able to go through. So the starch stayed put in our cells, and the iodine went through the bag into the inside. So we got the color change on the inside. But these silly students, since they put their starch on the outside and the iodine on the inside, that means the iodine is going to move out, and you'll see the color change outside of the cell. Moving on to question number 2. Up here in the Northeast, we certainly salt and salt and salt and salt our roads to get rid of that ice, and this does cause big problems. So let's think about osmosis and what happens in osmosis, and how can we apply our knowledge of osmosis to the salting of roads and the effects that that's going to have on the organisms near the roads. Alright, well, does salting the roads set up a hypo or hypertonic environment? So since it's a hypertonic environment for the organisms that live around the road, what's that concentration gradient going to look like for a plant? Right, there's going to be a low concentration of water outside of the plant and a high concentration of water inside of the plant. And so, of course, just like those onion cells shrank up when we put them in the salt water, the plant cells are going to shrink up when we salt the roads. And so it's going to cause osmotic stress to those plants. And if you notice when you drive down the roads, especially in the springtime, you'll see that mostly the evergreens, right alongside the road, the evergreens kind of burn on the side of the road, and a lot of that has to do with the salt. Moving on to question 3 on the next page. People who wear contacts know all about why they shouldn't use distilled water to clean their contacts, or use even fresh water to clean their contacts, that they need to use a special saline solution. That saline solution has just enough salt to be exactly the same as the concentration in your cells. So it sets up an isotonic solution. That way when you put the stuff on your nice fresh eyeballs, those cells aren't damaged. They don't either lose water or swell up and gain water. They stay just the same. So whenever IVs are given to people, we take careful care in making sure that that's an isotonic solution. But here in this question, instead of giving people the isotonic saline solution, we're going to switch that out with distilled water. So what's going to happen to this person? Well, is distilled water going to be hypo or hypertonic? What does that mean as far as concentration gradient? Where is there going to be a higher concentration of water? Inside of the cell or outside of the cell? Well, when we put cells in hypotonic solutions rather than a hypertonic where they shrink because they lose water to the environment, these guys are going to actually take on water. So when we give the person an IV of distilled water, the cells in their blood are going to take on water and eventually pop if it doesn't diffuse quickly enough, doesn't dilute, quickly enough. So big problem. Moving right along to question number 4. In this question, we're dealing with freshwater organisms. Now, these guys aren't in distilled water, but freshwater organisms, microorganisms live in the freshwater, still are dealing with hypotonic solutions. So these freshwater microorganisms are constantly taking on water. We're going to talk about this a little bit later on today in class. But many of these freshwater organisms have structures called contractile vacuoles. These are like sump pumps. You can almost think of a freshwater organism as like a paramecium, as being like a leaky submarine, where water is constantly leaking into the submarine, and so that's be somebody who's like bailing out the submarine all the time. And that's what the contractile vacuole is. It's bailing out the cell as the water diffuses into the organism. And of course, what are we calling that diffusion of water again? Right, that's osmosis. That's the process that's causing the water to flow into these organisms. Now, we're going to take these freshwater organisms that are constantly taking on water, and we're going to chuck them into salt water, where, of course, now we're talking about a hypertonic solution. And we have to remember what happens to cells when we put them in hypertonic solutions. Right? Since in a hypertonic solution the cell is going to tend to shrink, it's going to tend to lose water to its environment, well, what's the sense in having a contractile vacuole if you're already losing water? Duh. Alright, and the last question. By now, I hope that you are seeing the trend here in these questions, that you need to know what happens to cells when we put them in hypertonic and hypotonic solutions. And you need to be able to explain why that's taking place. So as with all of the other questions, we need to figure out what the concentration gradient is. By now, you recognize when you put salt on cells, that hypertonic solution that's set up because of the salt water is hypertonic, right, causes there to be a lower concentration of water outside of the cell than inside of the cell. So there's your concentration gradient. And the water then diffuses from the inside of the cell to the outside of the cell, causing the cell to shrink. So the cells in your tongue, and the cells on the lining of your mouth, the cells in the lining of your throat, the cells that are going down your esophagus even, they start to lose moisture, they start to shrink. Your body detects this and says, go get a Mountain Dew. I hope that answers most of the questions, or all of the questions that you have on this lab. I really hope that that helps review and start getting you thinking about osmosis and hypertonic and hypotonic and concentration gradients and all of that good stuff. I hope it hasn't replaced your studying that you did on parts of the cell membrane and what they do, because that's going to be on the quiz next as well. So good luck, and I'll see you next class.